Hello, everyone, and welcome to this very first webinar that is organized by the European Society for Gene and Cell Therapy in collaboration with the Coalition of International Gene Therapy Societies. My name is Juan Bueren. I am the director of the Biomedical Innovation Unit at the CIEMAT here in Madrid, in Spain. And currently, I serve as president of the European Society for Gene and Cell Therapy. Federica, can you show our first slide? Okay, you can see in this slide that our society is governed by the ESGCT board, and the members of this board are shown in the slide. And also, as you can see, we are a really international society. Most of the members are European, but not only European. Many other countries and even continents participate in our society. And significantly, we have almost duplicated the membership of our society during the last years. So nowadays, we have a really good representation of students, more senior academic members, and also a very significant contribution of members from the industry. Next slide, please. Okay, during the last ASGCT meeting that we had in Baltimore, we had a meeting with the presidents of very different societies of gene and cell therapy. And here we discussed the necessity of having more and more activities conducted in collaboration among all these societies. Therefore, we agreed to organize multiple activities, and these webinars are part of the activities that we decided to organize all together. So what we decided is that each of the societies will organize one of these periodic webinars. And this first one has been organized by the ESGCT, and thanks to the initiative of my colleague Alberto Auricchio, who is the vice president of the ESGCT and also the scientific director of the TIGEM. So I will allow him to do the introduction and to chair this session. And thank you very much again for all of you to participate and attend this very first webinar organized by ESGCT. So Alberto, you are welcome to continue with this session.
Thank you very much, Juan, for your introduction, and I'm also very glad to actually start this, be part of this first e-seminar of the Around the World series. So you will see on the screen instruction on how to submit questions. We will have two talks today. You will be able to submit questions at the end of each talk, so there will be a short discussion after each talk. So the first talk will be given by Federica Esposito, who is a PhD student in system medicine in my lab at TIGEM. And Federica got her master's degree in genetics and molecular biology from the University La Sapienza in Rome. And then as part of an Erasmus scholarship, she spent also some training time at Ubrecht Institute in Utrecht in the Netherlands before joining my lab, where she's working on AAV-based platforms so that allow to overcome some of the challenges associated with this gene therapy vector. In particular, she will talk about a new genome editing platform. So Federica, the stage is yours, and you have 20 minutes, and we are eager to hear your results.
So thank you, Alberto, for your introduction, and good afternoon, everyone. Just thank to the organizer also for giving me the opportunity to present my work today. And as Alberto also previously mentioned, I will share with you some data regarding one of my PhD projects, which is about a safe and effective liver-directed AAV-mediated homology-independent targeted integration in mouse model of inherited diseases. So just as a short reminder, the AAV vectors are the gold standard tool used in vivo gene therapy, and this is due to their safe and effective profile. And in particular, in the case of liver-directed gene therapy with AAVs, is well established that a single intravenous administration of this vector is sufficient to convert this organ, the liver, into a factory to achieve systemic expression and secretion of therapeutic proteins into the bloodstream. To date, 3 different AAV-based gene therapy products have been also commercialized by that rely on for the treatment of coagulation disorders such as hemophilia A and hemophilia B, but also there are several other ongoing clinical trials that also rely on this approach. And just as a brief mention, I want to say one carried out by our institute at TIGEM for the treatment of mucopolysaccharidosis type 6, which is one of the lysosomal storage disorder. Among some of the exclusion criteria that can be shared by the different AAV liver-directed gene therapy approach, one in particular, this case relating to the MPS 6 clinical trial, is that among the 9 patients that have been treated, all of them are older than 4 years of age. And this is due to one of the features of the AAV vectors, that is their nature that is episomal. So these vectors are not integrating in the nucleus of the host cells, but they stay as episomal in the nucleus of the transfected cells, in this case, the hepatocytes. And this, in the case of neonatal liver, which is a very proliferative organs, during time with proliferation leads to the vector loss over time, and therefore also the loss of the therapeutic efficacy provided with the transgene driven by the provided by the AAV vectors. And these features of the AAV vectors prevent, therefore, their use from neonatal treatment that often are the preferred stage of intervention for some diseases, such as inborn error of metabolism. Now, in order to overcome this limitation, in our lab, we took advantage of a CRISPR-Cas9-based knock-in approach that has been described in 2016 by Suzuki and Belmonte, which is called HITI, so homology-independent targeted integration. Now, the 2 important features of this platform are that, first of all, this is a mutation-independent approach, because with one single product, as I will show you, you can treat different mutations in the same gene, for example. And also, very importantly, this platform is cell cycle independent. Therefore, it's able to target both dividing and not dividing cells, because it hijacks the non-homologous and join repair pathway of the cells. Now, in this specific case, we are going to target the mouse albumin locus because it has been already reported to be a safe harbor for integration, and also it is a very active locus into the hepatocytes. And in particular, we are aiming at targeting the intron 13 of the mouse albumin locus because we don't want to affect the endogenous albumin expression, and we want to provide the integration of a therapeutic transgene in the safest way. Basically, the AAV system relies on the use of 2 different AAV vectors, where in our case, one AAV is carrying the nuclease, which is the SPCas9, under the expression of a liver-specific promoter in order to restrict the expression of the nuclease only into the target tissue, which in our case is the small hybrid liver-specific promoter. The other AAV vectors instead carries the HITI donor DNA, where important features are constituted by the presence of the coding sequence promoterless coding sequence of a desired transgene to get integrated into the endogenous locus, and at its extremities, both the 5' and 3' junction, there is the presence of the guide RNA, which is the same as the one in the endogenous locus, with the exception that is provided in an inverted orientation. Now, Upon co-infection of the two AAV vectors in the nucleus of the same cells, the guide RNA from the ET donor vector drives the nuclease cleavage both at the endogenous locus and the extremities of the donor DNA. This generates double-strand breaks that activate the non-homologous stain joining that, while repair this damage, will also integrate the donor DNA at the target site. Now, if the integration occurs in the desired way of the integration, the flanking guide RNA site will be destroyed, and this will prevent a further cleavage by the nuclease. In this condition, then, the integrated transgene will then get expressed by the endogenous promoter, which again is the albumin promoter. In order to assess the efficiency of our strategy, we first decided to use as a donor DNA vector the promoterless coding sequence of a reporter transgene, which is the DS-RED, and to assess this efficiency in wild-type mice that were injected as newborn. 1 month after treatment, we sacrificed the animals and we harvest the liver, and we performed different analyses such as histological analysis and liver cryosectioning and fluorescent microscopy, as you can see here in these pictures, where we found about 50% of the hepatocytes that were expressing DS-RED as a result of the integration only in the group of mice injected with the HETI and the guide RNA sequence. Thanks to these results, we also perform a comprehensive molecular analysis, thanks also to the help of the group of Tony Katomen from the University of Fribourg, by which we wanted to particularly study all the different possible outcomes of the integration upon ET. And in particular, we took advantage of their technology called CASTI-seq, which in this specific case was used to identify the different integration events happening at the on-target sites. As you can see in this graph on the left, by next generation sequencing, we found different numbers of reads that are represented here. Their abundance is represented by the height of each peaks that were touching different regions of the AAV donor vector. In order to understand how much given number to these integration events and assess their frequency, we decided then to measure the height of each peak. And the measurement of these peaks is then represented here in this cartoon on the right. And in particular, I just want to focus your attention on the peak number 1, which is the same as the one present in the graph on the left, that correspond to the HETI integration event of the donor DNA in the desired orientation. With a different method, we also, that was occurring with 20% of the frequency of 20%. Then with the second method, we also wanted to deeply investigate the role of the integration, if the donor DNA was actually really integrated thanks to the HETI-mediated integration, or if instead this was an event driven by AAV, so ITR-mediated integration. In particular, we wanted to really assess if the presence of the inverted guide RNA sites at the extremity of the donor DNA are important to drive and promote these HETI-mediated integration events. In order to do so, thanks again to the help of the group of Tony Katomen, in collaboration also with Manuel Riel, we perform a long-reads sequencing based on the nanopore technology, in which we used 2 different sets of primers. In one case, so the 5' junction, we selected the primers able to amplify both the endogenous target locus up to the end of the HETI donor DNA in order to amplify the full length of integration event from the 5' junction, and the same also for the 3'. Upon the analysis of all the generated reads derived from this long reads analysis, we found that the majority of the integration events were actually HETI-mediated events. So the DNA was integrating upon its digestion and released from the ITRs, whereas only a minority of the analyzed reads were present in sequencing with a reminder to the ITR sequences. Moreover, we also wanted to investigate the potential undesired integration events, such as the one of the nuclease, that again, by using the CAST-SEC technology, was found to be occurring, even though at a very low frequency compared to the proper heat integration events, and therefore, in this case specifically, 3 times less frequently. Moreover, we then decided to go forward and to assess the efficacy of our system in 2 different mouse models that require stable expression of secreting proteins where the liver can be used as a target tissue. So as a first example, I will show you data that we achieved in a mouse model of hemophilia A, which is an X-linked recessive bleeding disorder due to mutation within the factor VIII gene that is involved in the coagulation cascade, so in the intrinsic pathway in particular, and where the classification of these different forms of hemophilia A are due to the factor VIII activity levels, ranking from less than 1% in severe hemophilia A up to 40% with the mild forms. We took one mouse model of severe hemophilia A, so less than 1% of activity, and there we adapted our AAV platform. In this case, different from the reporter transgene, our HETI vector contains a promoterless coding sequence of a B-domain deleted factor VIII variant that can fit into the AAV vectors. And our system was, again, systemically injected in newborn mice, this time hemophilic mice. Following time at different time points when we went to monitor and measure the factor VIII activity levels, we found therapeutic levels of activity at different time point upon AAV treatment in guide RNA affected mice, which was found to ameliorate the hemophilic phenotype, as you can see from results obtained in the assessing the bleeding time of these mice after treatment that were decreased and improved coagulation profile in guide RNA treated mice compared to the scramble negative control mice. As a second model instead, I'll show you data that we achieved in the mouse model of mucopolysaccharidosis type 6, which we are very familiar with also thanks to the preclinical study of the clinical trial ongoing that I told you about before. And again, this is a lysosomal storage disorder due to the deficiency of an enzyme called aryl sulfatase B that is involved in the degradation of glycosaminoglycans. Now, the absence of this enzyme results in widespread GAG accumulation where the urinary excretion of GAGs is considered the main biomarker of the disease. And this is also a multi-system progressive disorder that affects different organs, but does not impair the central nervous system. And the current treatment is all based on enzyme replacement therapy. As previously done in the other mouse models, we, this case, used as a HETI donor DNA vector the promoterless coding sequence of the human RSB, and mice were injected again in bitemporal vein injection, so systemically, as neonates, with high doses of AAV HETI. These mice were monitored for a period of 1 year, and importantly, in each analyzed time points, by performing analysis that allowed us to measure the serum active RSB present in the sera of these mice, we found supraphysiological and very sustained levels of this enzyme at each of the analyzed time points, whereas in the scramble, so in the negative control group, these levels were completely undetectable. More importantly, we also found that the urinary GAGs levels in treated mice were decreased and normalized over time up to the last time point of the analysis, so up to 1 year post-treatment. Importantly, given the high doses tested, we also had some concern about the potential safety of this platform, and therefore we performed at 1 year post-treatment in these liver mice, in this treated liver, some molecular and histological analysis. First of all, these that you can see here are molecular analysis that we perform in order to assess the specificity of our guide RNA. In particular, the CAST-SEC analysis, again done in collaboration with the group of Tony Katome and Emmanuel Riel, allows us to assess the specificity of our guide RNA, because this technology is used to detect the potential chromosomal aberration or translocation, again, driven by off-target effects effects of the guide RNA. And this green line here allows us to identify that our guide RNA was actually very specific because we could not detect any chromosomal aberration. With the second and alternative method, we also selected the top 10 predicted off-target sites, and we independently analyzed each of that in order to detect any off-target indels. And any of the, in none of the analyzed off-target site, we could find detectable levels of indels in a combination different from the one observed in the scramble mice. Lastly, since we wanted to be sure that these livers were actually normal and so free from any potential neoplasm formation, we sent this liver section to a certified histopathology that confirmed the absence of any neoplasia or, for example, hepatocellular carcinoma. And therefore, the livers of these mice were considered completely normal. Since we used the high dose in the first study of AAV vectors, we also performed a dose-response study in order to assess potential AAV efficacy with doses that are half a log or a logarithm lower compared to the previous one used, because this dose, if directly translated to humans, can be beneficial. And when we assess the RSB activity, so we analyze the sera of the mice, both in medium dose group of mice treated or low dose of mice treated with our AAV system, and we again found detectable and examples also of supraphysiological levels of the enzyme. And regardless of any of those used, we found in both cases reduction and normalization of urinary GAGs level over time. Lastly, we also wanted to assess if ET could be effective also in adult mice rather than neonatal livers, and therefore we use adult MPS VI mice that were injected systemically around 6 weeks of age with the lowest dose of the AAV vector used in our dose response study, and these mice were again monitored for a period of almost 1 year. And each of the treated mice with the AAV system show again detectable and supraphysiological levels of the secreted serum RSB enzyme, and also importantly had a reduction and normalization of the urinary GAGs, therefore amelioration of the MPS VI phenotype. So, in conclusion, neonatal AAV has been shown to be productive, so it results in about 50% of correction to the hepatocytes with the high doses tested. Also, full-length heated donor DNA integration occurs predominantly upon the ITR's cleavage from the AAV vector donor. Neonatal AAV is effective in 2 different mouse models in which the liver can be used as a target tissue, in our case hemophilia A and MPS VI. And also it is safe even if provided at the high doses tested, with no evidence of hepatocellular carcinoma development or potential off-target effects. Of course, this is not the final setting, so in further perspective, we are planning to validate guide RNA sequences that are specific for the human albumin. Therefore, this will really improve the translational approach. And also, we will consider alternative delivery system for provided transient expression of the nuclease, such as non-viral vector, in order to limit its expression over time. And with that, I'll conclude. And first of all, I want to acknowledge my mentor, Alberto Ricchio, for giving me the opportunity to work in this project and also in many others. All the Auricchio's lab members, in particular our former member lab, Fabio Dell'Aquila and Stefano Auricchio, for really active work into this project. All the external collaboration, in particular Tony Catome and Mauro Elier for their huge effort put in the molecular analysis, and all of you for your attention.
Thank you very much, Federica, for a nice talk and also for being on time. I encourage the audience to submit the questions, and maybe in the meantime, Juan, do you have any questions?
Yes, sure. Wonderful talk. Congratulations. Very nice data.
Thank you.
So I have a couple of questions regarding your very nice studies. So if you try to do a comparison of the efficacy of the HITI approach in newborns and adults, can you obtain any type of conclusion, particularly not only in the short term, but also in the long term, whether treatment of newborns in this particular case is more efficient as compared to the adults? And the second one is, if you take into account normal doses of AAVs for the conventional gene therapy approach and the doses that you are using, do you need to use significantly higher doses to obtain a comparable clinical efficacy in the animals?
Okay, so thank you for your questions. So regarding the first part, we have done a kind of comparison between newborn and adults only if we take into account of the low dose tested, because we had performed a dose response study in MPS 6 mice where we used the same low dose of 6 per 10 to the 12 genome copies per kilo in newborn and also in adults. And actually we found in terms of improvement of the phenotype, similar outcomes, because both adults and newborn had improved phenotype of the MPS 6, so low GAGs in accumulation in different organs, such as liver, kidney, spleen, or the heart. And the phenotypic improvement also in the bones that I didn't have the time to show you in neonates. In adults, we also got a similar level of improvement, similar improvement in the different organs in which the GAGs are usually accumulated, so liver, kidney, spleen, whatever. What we did not really reach was the normalization in the bones, probably due to the already late stage in which we are intervening. But what I also would like to mention, this is maybe also related to the second part, is that while in neonates, after 2 weeks, we already see a decrease in actually an absence of the AAV vectors, therefore they are completely lost, and in this case also the nuclease is lost. In adults, we still have the AAV vectors are episomals, and therefore the nuclease is still active. Therefore, if at a certain time points, for example, after a month, we achieve a similar level of correction because of integration, let's say, because for example, using the DS-RED in newborns, we found about 2% of hepatocytes corrected. In the adults, we already after 1 month with the same doses, we found 5% of the hepatocytes corrected, but there the nuclease is still active. Therefore, I believe that we think that over time the vectors are still present, and therefore also the HITI can still be productive, let's say.
Thank you. Thank you.
We have 2 questions from the audience. Please, short answers.
Sorry.
So the first question is whether you have compared the HITI to HDR-based strategies, and if you compared what is the efficiency and the potential byproducts.
Okay, so we did not made a fair comparison between the HDR and the HITI, but what I can say is that we have several advantages using the HITI platform versus the HDR, because first of all, we do not require long homology arms, and therefore this, in terms of a kind of bioproduct that we can propose, allows us to incorporate longer transgene coding sequence compared to the one that might be used in the HDR approach. And also importantly, our approach is cell cycle independence, and therefore the same program with the same product we can treat either newborns or adults, and this will allow us to cover that portion of patients let's say patients that cannot be treated with the HDR, for example.
Thank you. The second question from the audience is when you talk about in future using non-viral methods, to what kind of methods do you refer, or what methods are you interested in?
So mainly let's say the most widely approach so far is the use of lipid nanoparticles, for example, to deliver the nuclease, while at the moment we think that the AAV vector, the donor DNA will be still provided with the AAV vector. So it will be an hybrid approach in which the nuclease will be driven by the LNP, and the second, the donor DNA will be driven by the
When you say the nucleus, what do you mean? What do you mean?
nuclease, like I would say, for example, Cas9 protein as a mRNA.
mRNA. Okay. Okay. All right. So there are no other questions from the audience, and I think time's up actually. So thank you very much, Federica. It was great. So we can move to the next talk. Will be given by Ivana Trapani. Ivana is currently a group leader at Teleton Institute of Genetics and Medicine, and also assistant professor at the Federico II University in Naples. Ivana got her master's degree in pharmaceutical biotechnology from the University of Naples, then did a PhD in my lab in molecular medicine. And then after a short stint as a postdoc, she was promoted to junior PI and group leader at TIGEM. And her primary research focuses on developing methods to overcome the limited cargo capacity of adeno-associated viruses. And I think this is going to be the topic Ivana will discuss during her talk today. So thank you, Ivana. The stage is yours.
Yes, thank you, Alberto, for this introduction. And good afternoon, everyone. It's my pleasure to be here today, and I want to thank the organizers for giving me the opportunity to present you today the results of this project we have been carrying out over the last years in the lab, which focused on the assessment of the efficiency and efficacy of a gene therapy approach, a novel gene therapy approach that we have developed for treatment of Stargardt disease in an animal model that we thought is relevant towards clinical translation. And so actually we developed over the course of the project a novel large animal model of our disease of interest, as you'll see in the presentation. So just a few words on the disease, that is Stargardt disease type 1. So this is the most common inherited juvenile macular degeneration with a frequency of 1 out of 10,000. It is inherited as an autosomal recessive disorder, and is characterized by the loss in the central vision, which occurs early in life, in the first or second decade, and is due to the progressive bilateral atrophy of the retinal pigment epithelial cells and photoreceptors, which occurs in a specific area of the retina, that is the macula. That is the region where there is the highest concentration of cones photoreceptors, which are the type of photoreceptors highly relevant for humans, for primates and for humans, because are those type of photoreceptors involved in daylight vision and color perception. The gene causative of Stargardt disease has been identified long ago, and it is ABCA4. This encodes for an ATP binding cassette transporter, which is located on the photoreceptor's outer segments, and specifically on the disc, on the rim of the discs of the photoreceptors. And here it is involved in the recycling of the visual chromophore. So just in few words, when the light hits the visual chromophore, the 11th retinal, this is trapped inside the discs, and ABCA4 works as a flip pace to translocate this compound outside of the disc, where it becomes available for the additional enzymes involved in the recycling of the visual chromophore and in the RP. So in the absence of ABCA4, or when this is dysfunctional, what happens is that this compound starts, the whole transretinal starts accumulating inside the discs and form compounds, toxic biocompounds, as the A2PE, which accumulates first in the outer segment discs and in the outer segments, and as the result of the daily phagocytosis of the photoreceptor's outer segments from the underlying retinal pigment epithelial cells, these compounds accumulates also in the RPE cells, under the form of lipofuscin granules that are these black dots that you see in this picture that is from the animal model, but it's the same in Stargardt patients. And these lipofuscin granules are toxic over time, so lead to the degeneration, the death of the RP cells and photoreceptors. So being a recessive disease, Stargardt disease is a suitable disease for gene therapy. However, there are some limitations in the gold standard vector for gene therapy, that is the adeno-associated viral vectors, which also Federica mentioned before, the AAVs, which limits development of gene therapies for Stargardt disease. And I don't need to introduce to this audience this vector that is the leading platform for in vivo gene therapy with the several AAV-based products already available on the market, but I just want to remind you that one of the main limitations of this vector is that they have a small packaging capacity and they can package only coding sequences which are below 4 kilobases. And this is clearly insufficient to deliver the coding sequence of several genes causative of inherited retinal disorders. And among these is also ABCA4, that with a coding sequence of 6.8 kilobases, of course, largely exceeds the AV cargo capacity. So to overcome this issue, we recently developed a novel approach, AAV-based approach, which is based on the process of protein transsplicing. So just a few words on the process. That is the protein splicing process is a process through which there is the post-translational conversion of a precursor protein into a mature protein through the removal of an internal element that is called the intine, that is capable to self-excise itself from the precursor protein, leading to formation of a peptide, the native peptide bond between the two flanking polypeptides, and so to the reconstitution of mature protein. Now, intines are protein domain widely distributed across different organisms in nature, and contiguous intine work exactly as I just showed you, but there are also split intines that are instead intines split into different halves, which are fused to two different precursor polypeptides, but they still retain the capability to recognize each other, assemble, and mediate the formation of the peptide bond between the two flanking polypeptides to this process that is called now protein transsplicing, because it occurs in trans between two different polypeptides. And still the result is a seamless mature protein, and this occurs without the need of any cofactor or energy source. So we decided to take advantage of this very powerful process to develop, to overcome the AAV limited cargo capacity. And we developed this approach, which we named the AAV intine, where the large BCA4 coding sequence is split into two halves, which are independently packaged into two AAV vectors. And in a cell which is confected by both vectors, the two halves of the protein are produced, fused to intines, which then mediate through the process of protein transsplicing, the reconstitution of the full-length native ABCA4 protein. And we tested this approach in several models, and I'm showing you just probably the most important data, where we prove that the levels we achieve with this approach is therapeutically, the levels are therapeutically relevant, because in ABCA4 knockout animals, we can reduce, or actually we can prevent the accumulation of lipofuscin in the RP cell layer. As you can see from these pictures and from the quantification, you see the dots, lipofuscin dots in the BC4 knockout animals, which are reduced in the AAV intine treated animals. So we prevent lipofuscin accumulation on this model. Now, having this promising data in our hands, we thought that it was important to test the efficiency of this platform in an animal model which was more relevant towards clinical translation, because the mouse model has some limitation. Indeed, while it recapitulates the accumulation of lipofuscin in the RP that is also found in Stargardt patients, ABCA4 mice do not show degeneration of the photoreceptors, so death of photoreceptors, which instead the Stargardt patient have until the end of their life. The photoreceptors never degenerate, and also they don't have any reduction of the retinal, any impairment of the retinal electrical activity. One hypothesis is that this is due to the different retinal architecture of the mouse retina, because as I mentioned before, humans have the macula where there is the high concentration of photoreceptors, while In the mouse retina, cones are distributed, they're less abundant throughout the entire retina. So to find a better model, large animal model, we decided to work on pigs because recent advances have allowed the creation of transgenic pigs in an easy way. And of course, especially for the eye, pigs are highly relevant because they have the retina of the pig retina is a site similar to humans. And also, most importantly, the pig retina has a high number of cones concentrated in a streak-like region where there is an high cone-rod ratio, so which resembles the macula. And also another important aspect is that the procedure that we can use to deliver gene therapies to the pig retina is highly comparable to the one that we use in humans, differently from the procedure that we use in mice to deliver gene therapy that is very divergent from the one that we use in humans. So for all this feature, we thought pigs could be a highly large animal model in which to test our gene therapy. So to generate a pig model of Stargardt disease, we engaged a collaboration with a company, Avantea, and with them we developed the BCA4 knockout pigs through somatic nuclear transfer. So specifically, we used the CRISPR-Cas9 technology to edit the BCA4 gene early in the coding sequence in the pig fibroblast, and then we characterized the clones and those showing mutation predicted to result in knockout of a BCA4. The nuclei of these clones were used to perform somatic nuclear transfer in pig oocyte, then induce blastocyst formation and implantation into recipient mothers, which then gave birth to a BCA4 knockout pig, so full germline BCA4 knockout animals. So we generated several litters, and we got in total of 12 pigs, 9 of which we used for characterization of the phenotype development over time. So as you can see from this summary table, we use different clones which carried different mutations in the BCA4 gene. And we kept the animals up to 15 months of age. That was the latest time point we could bring these large animals. And so first of all, in all the animals, we confirmed the genotype, the genotyping that was indeed as expecting, recapitulating the one of the clones, but also in the pigs, we could perform nanopore sequencing, which interestingly highlighted that beside the small deletion that we already identified in the clones, in some clones we were also having very large deletion in the BCA4 coding sequence, in the BCA4 gene, up to 2 kilobases, as expected by the use of Cas9 that we now know can also induce very large deletion. So we could assess a novel type of rearrangement in the BCA4 gene in these pigs. Independently from the mutation, that of course we selected the clones based on the assumption that the mutation were resulting in premature stop codons or defect in the mRNA, so abolishment of a BCA4 protein expression. And we confirmed that this was true in pigs because in the retinal lysets from all the different clones, we confirmed that there was undetectable BCA4 protein. So we confirm that these are BCA4 knockout pigs. So next we moved to evaluation of the phenotype of these animals. And first we looked at lipofuscin accumulation. And lipofuscin is an autofluorescent material. So we looked by fluorescence microscopy, and you can clearly see that the BCA4 knockout pigs have a massive accumulation of lipofuscin, that is this red signal that you can see here in the retinal pigment epithelium that is instead absent in wild-type animals. And interestingly, when we assess the lipofuscin accumulation and the distribution of such lipofuscin accumulation across the retina, and we analyzed we divided the retina in 4 different regions, in which region 3 is the one corresponding to the streak-like region with icon concentration. We interestingly found that indeed this is the region which has the highest level of lipofuscin accumulation. It also increases over time. This starts at 8.5. This is the earliest time point that we observed, 8.5 months of age, and increase over time. And I think you can see, I hope you can see this also in this picture where you can see knockout animals in region 3. We have the most intense lipofuscin accumulation. And also we quantified with a different test we did in collaboration with Rob Molday at University of British Columbia, where we, through HPLC, measured one of the main components, the compound I mentioned before of lipofuscin, it is the A2E. And so via HPLC, we confirmed that knockout animals have very high levels of lipofuscin accumulation, A2E accumulation in the RP. Next, we looked at the retinal degeneration, sign of potential retinal degeneration. And we did so by either evaluating the ONL thickness, the thickness of the outer nuclear layer, which is the layer which includes the nuclei of the photoreceptors. So degeneration, that photoreceptors that results in thinning of the ONL. And both on the DAPI evaluation fluorescent sections, but also on hematoxylin eosin stainings, unfortunately, we could not assess any clear retinal degeneration ongoing in these pigs, at least until 15 months of age. That is the last age that we evaluated. However, interestingly, when we tried to look at the retinal functionality in these pigs, we found, we performed the electroretinograms, so the test to assess retinal function in different light conditions. So in light condition, that it's a setup where we can evaluate mainly the function of cone photoreceptors, or in dark condition where we mostly evaluate rod function, or in condition where we can evaluate both rod and cone functionality. And interestingly, we found a significant reduction in the functionality of cone photoreceptors in these animal models, suggesting that this can be a preliminary sign of the dysfunction of photoreceptors, that although we don't have any retinal degeneration yet, photoreceptors are starting suffering because of the lipofuscin accumulation. So having in our hands this promising model, we decided to look to evaluate the efficacy of our gene therapy platform, the gene therapy platform that we developed. And we used the 3 ABCA4 knockout pigs of 2-3 years of age that were the oldest that we generated. And we injected either one eye with our AAV-intent vectors, or the other eye we left it, we treated with the formulation buffer as negative control. And we co-injected the 2 AAV-intent vectors along with a 10-fold lower dose of AAV vector expressing the reporter GFP protein to track the transduced area. We performed 2 blebs in the streak-like region of the retina of the pigs and harvested the eyes at 2 months post-injection. And the first, we looked at the expression levels, and as you can see from this Western blot analysis, we achieved very high levels of ABCA4 protein reconstitution in ABCA4 knockout animals, which ABCA4 was otherwise absent in the knockout treated with the formulation buffer, and these levels were found to be comparable or even exceeding the endogenous ABCA4 levels that you can see in wild-type animals that are the ABCA4 proteins that pointed by the arrow, the orange arrow in this Western blot. So these were very high levels of ABCA4 protein expression. And so next we moved to evaluate the efficiency of our approach. And as you can see, both by the images and by the quantification in the AAV-intent treated eyes, we could get a reduction in lipofuscin accumulation that was significant in the youngest, in the animals that were treated at 2 years of age. And this result is particularly important for us because this is a setting which we think it's closer to the setting of Stargardt patients, differently from the mouse studies, because as I showed you before, in knockout pigs, we know that the lipofuscin accumulation is already ongoing since at least 8.5 months of age, and now we are treating them at 2-3 years of age. So there is a massive, we expect in these mice to have, in these pigs to have massive lipofuscin accumulation. While in the mouse studies, we could only assess prevention of lipofuscin accumulation. So this is a situation that is closer to the Stargardt patients, which we expect to have already massive lipofuscin accumulation ongoing by the time they reach the clinic. And also importantly, this was only 2 months after injection, and considering the time that AAV vectors needs to express and the protein transfer splicing process needs to take place. So this is really a short timing upon injection. So we think these are really relevant data. So to conclude, I've shown you that we developed a large animal model of Stargardt using the CRISPR-Cas9 mediated editing of ABCA4. These ABCA4 knockout animals show increased lipofuscin accumulation in the retinal pigment epithelial cells, the main hallmark of Stargardt disease, and preliminary sign of impairment of the retinal electrical activity, although unfortunately in the absence of marked retinal degeneration, at least until the 15 months of age that we analyzed. And in this animal model, we tested for the first time the AAV-Inten platform. For the first time in a large animal model, we tested the AAV-Inten vectors, and we proved this platform to effectively restore ABCA4 protein expression, resulting in improvement of the retinal phenotype in this animal model. And so we think that our data further support the clinical translation of this novel platform, the ABCA4 Inten vectors in Stargardt patients. So to conclude, I want to acknowledge all the people involved in the studies, Eugenio Pugni and Federica Trapani from my lab, especially Alberto Auricchio and the people in his group, which also contributed to this project, the other collaborators in TIGEM Bioinformatics core, our external collaborator Avantea, which generated the animal model, the University of Bologna, which helped us in the handling and the injection of the pigs, the group of Professoressa Simonelli at the University of Campania, Luigi Vanvitelli, that also helped us in performing all the analysis and the injection, and our collaborators at the University of Leeds and University of British Columbia that helped us in the phenotyping of the animal models, all the funding agency, and all of you for your attention. Thank you very much.
Thank you very much, Ivana. This was rather clear presentation, but of course I know about this. Let's see if there are questions from the audience. No questions from the audience yet. So maybe Juan, do you have?
Sure. Yes. Very nice work. Incredible animal model. I can imagine it was not an easy task to develop the animal model, and also the gene therapy approach is really amazing. So regarding the animal model, Ivana, I wonder whether the animals that you generated have the knockouts in whether these were monoallelic or whether many of them have the knockouts in both alleles. And also I wonder whether you had a chimeric population of genetically modified and non-modified cells. And also I wonder whether this genetic modification took place in the germ cells, and therefore whether the genetic modification was transmitted to the descendancy.
So yeah, so the mutation, I'm sorry whether if this was unclear, maybe the process that we used. So the genetic modification is biallelic. So we confirm that we have both alleles mutated. Actually, we selected those, so the procedure worked so that we modified the genome of pig fibroblasts. And so we selected only the clones showing biallelic mutation. And these are all basically compound heterozygous mutation that we isolated. The nuclei of these clones were used to generate the oocyte, and so to generate the ABCA4 knockout pigs. So, but also we confirmed that the same genotype was found in the germline, in the litters of the pregnant sow. And so because of the process, this cannot be a genetically heterogeneous population. This is one nuclei that we moved into oocyte and induced formation of blastocyst. And so all the cells in the body have the same genotype. And indeed, we confirmed this because we did the genotyping on a piece of the hair, and it was the same genotype that we found in the.
Really nice, nice. Are there questions from the audience, Alberto?
Yeah, there is one. And actually, just to finish. I mean, and of course, the animals that have the genetic modification, because it's a germline modification, will transfer the modification to the offspring if you breed them. So so then you have two choices. You can either to have affected animals. You can either breed animals coming from this somatic nuclear transfer, or you can do somatic nuclear transfer from the original clones every time you want animals. So you can choose basically if you want a colony or just some animals for specific experiment. But going back to the audience question, the question is around the promoter that you used in your AAV vectors, Ivana.
Sì, so this was a photoreceptor specific promoter, the GRK1 promoter. So we are getting, we are basically reconstituting ABCA4 expression specifically in the photoreceptors. And I think, I mean, this is a very interesting data also what we got, because there is a little bit of controversy on the expression of ABCA4 that has also been proved to be at very low level in the retinal pigment epithelial cell. But I think that basically our data show that with the photoreceptor specific promoter, we can get improvement of the phenotype of the lipofuscin phenotype. So I think this was an important data as well to clarify the mechanism a bit.
And Ivana, regarding the transduction process with the two AAVs, so does this procedure imply that the dose of the AAVs that you have to use should be twofold as compared to conventional strategies? And also, I wonder whether in what percentage of cells you have AAVs from both types?
Yeah, so actually, so for our studies, we used a dose that is comparable to doses that are used also in large animal models, even for approaching using a single AAV. Also because we have the volume that is limiting, so I mean, we cannot go so much above that dose. But the important aspect is that actually I think we clearly proved in several studies that we don't need to go so much with a dose that is so high, because in the eye we have very high co-transduction rates. We proved in different animal models that we can have above 75 in different large animal models, including the pigs, that we have above 75% of photoreceptors that co-express, that are co-transduced by both vectors. So the doses we use are basically the same that are used with single AAV vectors. We are not really increased that a lot.
Very nice. Thank you. Thank you.
So there is a second, let's say, a corollary question to the original of the promoter, which is if the photoreceptor-specific promoter that you used was the same that you used for the GFP vector, the vector to track the injection site.
Yeah, so yes, we used the same promoter and the same also AAV serotypes, just at a tenfold lower dose. And the reason why we did so, because we didn't want to use a, since we were looking for a phenotype into the RPE cell layer, we didn't want to have GFP in the RPE cell layer that could interfere with the measurement of lipofuscin, or you know, might add up to the toxicity of lipofuscin in the RPE cell layer. So we stick to the photoreceptors layer.
Sure. Okay, I don't see additional question from the online audience. We've been extremely precise. It's 2 minutes to 3. So I would like to thank you very much, both Federica and Ivana, for taking the time to give these 2 talks. I would like to thank Juan for introducing the session and being with us throughout the whole session, and of course Lumina for helping organizing this seminar. And I hope this is just the start of a new series of seminar, hopefully with a large online audience. Thank you very much, everybody, and see you at the next e-seminar.
And even in Rome.
And even in Rome at the end of October for our annual meeting. Be there.
Bye-bye, everybody.
Bye-bye.
Bye. Thank you.
Bye.